Yep.
All right, thanks. All right, as stated, I'm Matthew Hansen. I'm a scientist at the Nanotechnology Characterization Laboratory. Today I'll be talking about ICP-MS for elemental analysis of cancer nanotherapeutics. And so today I'm going to discuss kind of three things. So first I'm going to give an overview of the NCL just to familiarize the audience with what we do here and the services that we provide. After that, I'm going to talk about how we use ICP-MS. We like to use ICP-MS for pre-screening these nanotherapeutics as we get them in. We also use ICP-MS in animal studies to help us track these nanoparticles as they pass through the animal. And then finally, I'm going to talk about some single particle work that we're doing for individual particle measurements. And so I further split this talk up. I'm going to look at 5 different nanoparticles and kind of highlight each of these points using different nanoparticles as I talk today. So the Nanotechnology Characterization Laboratory was established in 2004. It's a collaboration amongst the NCI, NIST, and the FDA. And we have 4 primary objectives: to characterize nanoparticles using standardized methods, to conduct structural activity relationship studies, to facilitate regulatory reviews of nanotech constructs, and to engage in educational and knowledge sharing efforts. And so how we do this is highlighted down here in this picture. So what we do is we accept particles from extramural community. So these could be from education, from small startup companies. They apply to our program and we review them through a white paper process. If we accept them, then what we do is run our assay cascade for them. And so we'll sit down and create a research plan with them and come up with things that we're going to test with them. And it's really, we like to stress that if you're accepted into this program, all these studies and stuff that we do are free of charge for whoever's accepted. And so it's a great resource when trying to get these cancer therapeutics into clinical trials and clinical settings. Furthermore, our assay cascade is split into 3 predominant sections. I am part of the physical chemical characterization section. And so that's what I'm going to be focusing on today. And what we do in this section is look at size, composition, purity, surface chemistry, and stability. And so we have many different instruments that we can actually look at all of these things. And again, today I'm just focusing on ICP-MS, which we can use for compositional analysis and for animal studies and a lot of other things. And so we like to support these collaborators and trying to get them into clinical studies. So that's our ultimate goal, is to get these nano cancer therapeutics from benchtop into clinical studies and hopefully out as a product. And so far, as of today, we've gotten 14 collaborators into clinical trials, and that they're shown down here below. If you would like any additional information, you can actually visit our website. We have a lot of good information there. It also tells you how to apply to the program. We have a lot of standard operating procedures and methods development up there as well for many different instruments. And so please visit there. We also have some contact information. So if you have any questions, you can get a hold of someone at the NCL if you need to. So sticking with the NCL for a little bit, over the last 3 to 4 years, we kind of see some trends in nanoparticles. And so our most recent trends here, we see about 20% of the nanoparticles that we receive are inorganic nanoparticles. So these are metal, silicon, or iron based. And so this is obviously where we use ICP-MS, but we use it well beyond traditional inorganic molecules. So recently we were getting a lot more nano drugs and nano cancer therapeutics that have elements that are part of the drug itself that we can actually track and quantitate via ICP-MS. And so it's become become equally useful or more useful outside of the traditional metal nanoparticles as it is within them. And so I will show that with a few examples today as well. In addition, as these polymer nanoparticles start growing in the size that we've been getting, there's also a lot of metallic impurities that we really need to test for that are due to residual catalysts and other things that we need to know that's there before we can actually introduce them into in vitro and in vivo trials. And so just finally here, these are just some of all the elements that we've actually looked at here that's been part of some nanocancer therapeutic, and it's quite a bit and ranges from many different properties. I further highlighted some of them in red, that's iron and gold, and these have by far been the 2 most abundant metallic elements that we've actually gotten as far as application-wise. So moving on. So ICP-MS is very important for our pre-screening process. And what pre-screening is we receive these particles and we want to first verify everything that our client says is there and make sure there's no impurities and nothing else that's going to interfere with any of our in vitro and in vivo studies later on down the line. And so to illustrate this point, I'm going to look at this particle 1. So this is a polymeric nanoparticle containing boron. And when we receive this in 3 separate vials. And so one of the first things we have to do is look at this nanoparticle and also make sure that each of these samples in these different vials are equivalent or the same, so that, again, we know what we're getting into. And to do this, one of the first things we like to do is run a semi-quantitative analysis on these particles. And it does 2 things for us. It helps us gauge our concentration range for the active drug itself. So in this case, in the upper left corner, I have highlighted in blue boron, which is from our active drug. This lets us kind of gauge where we need to measure this at and help us really fine-tune our calibration range for that, as well as it helps us look for impurities. So if we look to the graph on the top right, I've highlighted in red copper and ruthenium, and we saw quite a bit of those in this sample. And it turns out that these are catalysts that were used to create the polymer that are just residual metals that weren't filtered out or cleaned out from the sample. And so when we went to quantitate the amount of boron in the sample, we were given an approximate drug content by the client, and it's shown down here. There was about 133 micrograms per mil. When we measured boron, we actually see about half that in samples. And so from this, we know that our drug content is a little bit lower than it should be, and that we do have residual catalysts that we actually need to now quantify and look at. So when we looked at each of these individual vials, A, B, and C, we wanted to know, are these equivalent? So the first thing we did to do this is just quantify these 3 things, so the boron, the ruthenium, and the copper. And the boron for vials A and B were fairly similar. And so we think that these are pretty much equivalent samples. And if we look at the catalysts for all 3, they're pretty similar. When we compare the boron for vial C to A and B, it's actually quite a bit larger. And so we wanted to look at the ratios between these 3 elements to see if these are just, if the samples are the same and just maybe a concentration thing moving through, if there is something different. So when we compare the 2 catalysts to boron for A, B, and C, we see that A and B are similar for the 2 catalysts to boron ratio, and then the catalyst to catalyst ratio is also very similar. When we looked at C, we saw that this is actually quite different now for the catalyst to boron ratios, that they're much lower. However, the catalyst, the copper ruthenium, when you compare the 2 catalysts to each other, they're actually a similar ratio to the other 2. So from this, we were able to conclude that A and B are most likely equivalent samples, and that C is substantially different. So we don't know why this is. We didn't further investigate this, but we just know that it's not equivalent. So moving on, we're going to look at a second particle now, and this is a liposomal nanoparticle, and this one contains copper. And so there is some concern that not all the copper was staying within the nanoparticle. And so the client wanted to look at free versus drug loading of this particle. And we did this by separating our sample out. And to separate it, we use these microcentrifuge tubes. So we basically put the sample on the top. There's a membrane. In this case, we used both 100 kilodalton cutoff and a 10 kilodalton cutoff. And then we centrifuge it, and then that allows the liquid to pass through, and then we can retain the nanoparticles at the top. We collect both, and we run ICP-MS on them. And so we ended up running 2 different sized membranes for this, just because we wanted to ensure that we weren't extruding some of the nanoparticle sample through the membrane and skewing our results a bit. And so when we look at both the 100 kilodalton and 10 kilodalton results, we get the same results. We get about 40 to 45% is actually free copper, and then 50 to 55% is actually retained within the nanoparticle. And then in the bottom corner, when we went and did the semi-quantitative analysis, which we like to do for all nanoparticles, we actually see that there's no contaminants, and the only component in there is actually copper. And so we were able to answer this question for our client and to show that the copper is most likely exhibiting an equilibrium between solution and nanoparticle, just because it's around 50% for each. And so once we've done all of these pre-screens, these nanoparticles get used in animal studies. And so I'm going to talk about some animal studies next and show you how ICP-MS is actually very useful for us for these. And to look at these animal studies, we're going to look at another particle. So this is a polymeric nanoparticle containing iodine. And this is about 30 nanometers in size, this particle. So what we noticed with this particle is extremely stable. And so we had to actually pre-digest this sample. And you can actually see in our concentration chart when we were quantifying the amount of drug that we're going to use for this animal study in our dosing solutions, we see that if we don't microwave this sample, we actually only see about a third of the total iodine. When we pre-break down this sample, we see all of it, and it's in agreement with our client. And so we just like to, just make sure that when you're working with nanoparticles, they are small, they are suspended, but sometimes you really have to make sure that you're really working up the samples when necessary. And I'm showing down here that we have a control recovery, so we have the unconjugated drug that we can also look at. And regardless of whether we microwaved or not, we were constantly getting 100% recovery. So that kind of, could throw you off as well if you don't really take these steps beforehand. And so this nanoparticle was used in an animal study, and they wanted to look at clearance. And so they were going to do this looking at 24 hours clearance through waste, so through feces and urine, and then through 17 days to see how much particle is actually still circulating in the animal. And we have 3 groups for this. We have our control group, which is just a blank, so they're administered saline. I like these samples particularly because they allow me to matrix match our standards. And so this will help reduce any discrepancies that may be caused because of matrix issues, especially in high matrix samples like feces, where you have a lot of stuff going on. And then we have a high and a low dose group. And again, they're collecting these excrements at 24 hours and 17 days. And what really we'd like to highlight with this is that our high dose and low dose, we could easily quantify after 24 hours. And even at 17 days out, there was ample iodine that we could actually still probably quantify iodine even more days out if it's still within the animal itself, because ICP-MS it's so sensitive that we can actually see these, whereas I think we would have missed it with some other techniques. So using this particle, we actually had another animal study as well. And so our client was concerned with 2 things. One, they want to know if this nanoparticle is going into the brain. And for them, that's really good news if it is. That's what they're trying to do. They're trying to get this particle into the brain to help image and treat brain cancers. And I just want to know how this, if this nanoparticle is being excreted and how much. And so we were giving 2 samples from multiple animals. It's brains and then these clearance organs that are kidney, liver, and spleen. And so when we received these, the brains themselves were in 15 milliliters of formalin, and then the kidney, liver, and spleen were all combined into one Eppendorf tube of 50 milliliters of formalin. And we received these, and they were sitting around while waiting to be analyzed, just because we had some staff turnover and I hadn't started yet. So these were sitting around for quite a while. And so we were actually concerned that because they were in so much liquid that we may be leaching out some of the nanomaterial and we may be actually losing it into the formalin. And also with the clearance organs, we were also concerned that, if we are leaching these nanoparticles out, it could be commingling and giving us false results with some of these organs if there wasn't supposed to be any drug in them. So what we ended up doing was quantifying both the tissue samples and the formalin just to answer these questions of how much is going to be leaching over time, and is this going to be a concern. So first and foremost, if we focus on the organs themselves, we actually do see quite a bit of this drug through quantitating iodine in the brains themselves, and that's really good, and it was great news for our clients, and they liked hearing that. And as well as the organs, we see quite a bit of it just accumulating in the organs. And we don't have any time studies for this. We don't know how fast they're cleared or what. These are just single point collections. But when we do look at the formalin, we actually see around 2.5 to 5% of iodine in the formalin compared to what tissues were in there. And so this could be substantial, especially if an organ doesn't have any in there and some of the other organs have high amounts of this drug in it. You could get some cross-contamination. So we always just like to harp that it's really important how you treat your samples even before we get them, because this instrument is very sensitive and your results are only going to be as good as the storage conditions and how we receive the samples. And just to highlight how sensitive this is, we were able to quantitate iodine down into the single parts per billion range. And so we have plenty of leeway with this as well as far as sensitivity. And so for the final section, I'm actually going to talk about how we use single particle ICP-MS and what we've been doing so far in the lab with it. It's not quite as extensive as some of the other stuff we do, but it is something that we think is going to be continually to be important for us, and I can explain a little bit why. So for the single particle analysis, we're going to look at two things. One, we're going to look at concentration determination, and then later on, I'm going to talk about some compositional analysis that we did using it. So for this first part, for the concentration analysis, we're going to use pegylated gold nanoparticles. And so we're using the pegylated gold nanoparticles because we want to cross-compare these nanoparticles with single particle ICP-MS and some of the other instruments that we have that do particle concentration in our lab. And pegylated gold nanoparticles are a little bit more stable than the citrate-coated counterparts just because there's an actual chemoabsorbed surfactant on there. And some of these other techniques actually will use high ionic strength, which citrate stabilized gold doesn't really like well. And so before we can actually start comparing these instruments with each other, we have to understand our solution that we're going to be using for this. And so what we have here is our pegylated gold nanoparticles, and we looked at these using single particle counting ICP-MS, and we did this three times basically. So the gray and the blue in the chart are just your standard if you're using the nano application for the PerkinElmer, using a 50 microsecond acquisition time, so we can raster across the entire nanoparticle peak. And this is the gray and the blue were just taken on 2 different days to show that there is consistency within our results even across multiple days. And so we see that our diameter is fairly close to the reported diameter from Ted Pella, which was a supplier that we coated with PEG. And the concentration is fairly close. It's within an order of magnitude. It's within, a couple tens of percentage of what we calculated based on the total gold content in the solution. And then highlighted in yellow is using single particle counting, but using a much longer integration time, so a 10 millisecond integration time versus this 50 microsecond. We like to do this because we do have an older ICP-MS as well, and that one that's the shortest acquisition time basically you can do is about 10 milliseconds. And so this is good because we can cross-compare an older instrument with a newer instrument, and just to see how stable this is across multiple different ways of testing it. And so we see very consistent results with this longer acquisition time that we saw to the shorter acquisition times. Our sizes are almost identical to the reported values, and the concentration is also very close. So when I was carrying out these experiments, we had some power issues where some of my runs got cut short because we had a power surge or the power went out. And when I was redoing all of these solutions to create very long calibration curves and look at stuff, I was interested in knowing that how the stability was because I kept having to redo this multiple times. And so, especially since single particle counting requires pretty extensive dilution of these particles, it's really good to know if they're going to be stable and how long are they going to be stable and what we should expect. And so the graph on the left is just concentration stability over time. So what we did was we made up these solutions, measured them on day 0, took the same solution, measured it on day 3, and then 7 days later. And over the course of 7 days, we actually see a consistent drop in the concentration. So we see a concentration drop of about 40% on average across all of these nanoparticles. But we can actually see the 2 particles, the 100 nanometer gold and the pegylated gold 100 nanometer silver and the pegylated gold nanoparticle actually seem to be a little bit more stable than the other ones. And they only decrease about 30% over 7 days. When we looked at size stability over this time, however, we actually saw a very different trend. We see that the size are fairly stable over 7 days and that there's really not much changing. And so what this told us is that nothing's really happening with the nanoparticles. They're kind of staying intact, but what's happening is as they're diluted, the surfactants releasing from solution, they're kind of crashing out. And that's affecting the concentration, but generally not affecting the size as much. And so we just like to say that if you're going to do concentration, fresh is always better, even for size or concentration, but you have a little bit more leeway if you're only doing size analysis with this instrument. And so moving on and to looking at these same nanoparticles and comparing our ICP-MS results with other instruments in our lab, we basically have two other instruments that we're looking at comparing concentration with the ICP-MS, and that's using light scattering, and resistible sensing, and then ICP-MS. So each of these are great methods for different reasons. For the light scattering, it's pretty easy. You dilute your sample and you run it, and it'll give you size and concentration analysis. The problem is you need a higher amount of concentration. So ICP-MS is actually a little bit better just because you can use lower concentrations and still get good results. With the resistible sensing, like ICP-MS, it's an individual sensing technique. This one uses a pore that they run a current across. As the nanoparticle passes through, you get resistance, and it shows up as a little blip, and that is basically your particle. And so similar to ICP-MS, you're just counting your particle as it passes through to get a concentration. You can also get size information, and you can also get speed as it passes through, and that'll give you some zeta potential information. Some of the drawbacks of this one are highlighted by why we're doing this. It's because there is no traceable nanoparticle standard solution. So we can't directly compare these to a standard solution and to each other because we just currently don't have one. And that's really why we're looking at how do we compare these instruments to each other. And that's really why this resistible sensing, they actually do need a concentration standard to get an accurate concentration from it. Whereas ICP-MS we like because it's very sensitive, it's very dilute, you can use it in any buffer you want, and you're only limited by how accurate your basically your flow rate is or your transport efficiency. And so here we're using a peristaltic pump, so I'm sure we could get actually more accurate ways of determining our flow rate using different sample introduction systems. But as long as you know accurately what your flow rate is, you can actually, without a nanoparticle standard, measure the concentration of these. So we think this might be a good baseline technique to then branch out and compare other techniques to. So if we look at our pegylated gold nanoparticles, these are very preliminary results, and we don't know why we're getting the results that we're getting yet. We're still looking into this, but just to show you that they are different, and this kind of really highlights the need for an actual traceable particle concentration standard and why we would need it. So if we look at our estimated concentration for the nanoparticles, about 4.5 times 10 to the 9 particles per milliliter. We see light scattering gives us about 3 orders of magnitude more concentrated particles than that. We see our resistible sensing, it's actually a lot closer. It's about an order of magnitude off. And then our ICP-MS is even closer to this value. And it's within the same order of magnitude. And again, this is an estimated concentration based on total gold content of this solution. So it could be a little bit different here and there. But again, we're just highlighting that, we're trying to find a way to compare these instruments, and really we need a traceable standard, but we actually also like ICP-MS and single particle counting, especially for metal nanoparticles, because it seems to be independent of any other fitting. You're just basically literally counting it, and as long as you know your transport efficiency, it's easy to get a concentration out of it. And so finally, we're going to look at one last nanoparticle, and this is for our compositional analysis that we've done here. And so this is a gold silica core gold shell nanoparticle. And so initially when we ran this using single particle counting ICP-MS, we ran it about 1 to 500,000 dilution. So as I talked about, these are some pretty big drastic dilutions that we run. We actually see what's shown on here in the pink bar graph. So we see a main peak that I've labeled as 1x, and then we see some other main peaks at about 2x, 3x, and then in the pink instance of the pink, we can see some 4 and some 5x. Usually when we see this through single particle counting, we're like, okay, our concentration, our sample's too concentrated. We need to dilute this down. What's happening is we're getting actually multiple particles hitting our detector at a time. That's why we're getting these whole number multiples. So that's what we did. We decided, okay, let's dilute this down, and let's see if this is really just multiple particles hitting our detector, or if this is something inherent in the sample. And so once we diluted these down, we actually still see that these multiple peaks show up. And as we continue to dilute down, the ratio of them remain fairly stable. So at this point, we're thinking, okay, so this is something in the sample, and we need to figure this out further. So what we did is we took this sample and we separated it out using centrifugal force field flow fractionation, which is a separation technique that's good for inorganic nanoparticles. And what we did was we manually collected 3 fractions. So if we look at the chromatogram up here, we actually see 2 peaks and a shoulder, and that's what we collected. So our first fraction we collected is this highlighted in orange, is that first peak. The second peak is just highlighted in silver, we also collected. And then the third, that little hump at the end, we also collected, and we highlight that in gold. And so then we ran single particle analysis on each of these 3 fractions that we collected. And that's what's shown in the graph down at the bottom. So the blue histogram that we see, that's our unfractionated, our whole sample. And we see the multiple peaks that we've constantly been seeing before. When we did single particle analysis on that first peak, we only see the 1x or the monomer peak. When we take it from the second peak, which was highlighted in silver, we only see the dimers, so only the multiple of 2x. And then that final hump is highlighted in gold, we actually see the 3x peak and some 4x, 5x out there. And then to further want to actually look at what's going on in the sample, we then took each of these fractions and ran TEM analysis on them just to kind of confirm what we're suspecting was happening. And that's shown here in the pictures on the right. At the bottom, highlighted in orange, is that first monomer peak, and what we see is pretty much just all single nanoparticles, no dimers, nothing else. The middle picture shows that we have a more dumbbell, so we have 2 fused particles together that have basically formed 1 particle. And then from the third fraction, we actually see the multimers. We see basically 3 particles plus just fused together and forming these multiple dimers, multimers. And so furthermore, we were given the blank silicon shell without any coating by our client. And we ran TEM on those just to get an actual size of the core. And then we helped use the size of the core to kind of, along with single particle counting, to then determine our shell thickness of each of these. And that's also shown in the pictures. So our monomers, we were able to determine we have a shell thickness of about 14 nanometers. The dimers were about 14. And then the trimers, multimers were around 12 to 13 nanometers. And with that, that concludes my talk. And I would like to thank my team at the NCL. They're all shown down here in the picture. It's a great group of scientists that we work with here, all very smart and talented individuals. And without their help, we wouldn't be able to do half of the stuff that we could do. And so I'd just like to remind everyone that if you have any more questions, feel free to visit our website at ncl.cancer.gov. There's a lot of useful information there. There's information on how to apply to our program. That we have some standard operating procedures and some protocols for a lot of different instruments on there that may be helpful if you're looking at nanotherapeutics. And as always, I'm glad to answer any questions you have. Thank you.
Well, thank you, Matt. That was a fantastic presentation. Actually, some really interesting research and work there. We had a few questions. I just wanted to remind everybody that you can type in your questions in the question panel of GoToWebinar. So I guess I'll start with the first one here, Matt. The first one is a question: what is the stability of the standards for your single particle? I think you kind of covered that, but
yeah, I kind of covered that. We like to use them fresh every day, and
yeah,
all the nanoparticles I actually showed were the ones I was using as the standard. The only one that I didn't consider a standard is the pegylated gold. But again, those are standards in the sense that they're off-the-shelf nanoparticles, so we have to kind of take them at. We try to do some gold analysis on them and then some TEM in order to kind of get their own concentrations as well, just because none of them have been actually standardized or verified.
So yeah, these are they're coming to you with a certificate of analysis, right? Though on the size, not usually on the particle concentration.
Yeah, so it depends. So the gold ones just give us a gold content and they give us a size. I guess there is a certificate of analysis. The nanoparticles we're using is like a nano. It's like a nano exact, so they're kind of more supposed to be a standard, I guess, but they're not anything. And so they actually do come with a certificate that tells us a concentration and a size, whereas the gold ones don't give us the particle concentration, they give us the total gold content.
They give the total gold in the sample. Yeah. I've been missing the NIST materials as well. They sold out pretty fast when people started getting into this. All right, so next question. Do you think the particles dropped out of solution over time?
Yeah, I think that's what was happening. So especially since these are citrate capped particles, as we're diluting these down, there's no, we dilute them just in water. And so there's nothing really keeping the citrate on other than electrostatic forces. And so as we're diluting these down, what we think happens is, the citrate slowly falling off these particles, and as such, they're becoming less stable. And so there's nothing keeping them in solution. So they just kind of fall out and stick to the sides or stick to each other and fall out over time.
Yeah, that's what I suspect as well. And then I guess there's a follow-up to that. Did you see any other evidence showing some of the particles not in solution? For example, precipitated?
No.
I mean, this is nano. It's tough to see.
Yeah, with gold, you can usually tell if they're precipitating out. You see
True.
some stuff floating in there, but in this case, I didn't really see any of that. And again, these are extremely dilute solutions. So usually in these, gold solutions are usually like red, and those still, even though you're seeing the red, they're still fairly dilute. They're not that concentrated. But these are even more concentrated, like almost can be upwards of 1 million times more concentrated than that. And they just look like a clear solution. So even if they are aggregating out and agglomerating or sticking together, it's going to be hard to see just because there's just not a lot in there.
Yeah, exactly. Okay, next question. What the wording isn't perfect, but what with the gold nanoparticle standards from LGC that is certified to particle content? I guess the question is if some of the gold nanoparticles from LGC are certified with particle content or concentration.
I haven't used those, so I don't know.
Yeah, I haven't used them either, so I mean, I'd have to look into that as well. I've seen attempts at selling particle standards with particle concentration on the certificate of analysis. So there are some available. They're okay from my work. Gold. Yeah. So from ICP-MS results, how would you be able to decide that a sample needs further dilution, whether 3 times or 5 times or whatever?
In regards to single particle analysis?
Yeah, I guess that's what the question's referring to, yeah.
So basically what we look for, like I mentioned during the compositional analysis, if you see peaks that are multimers of your main peak, that's kind of indicative that you're actually seeing you have a solution that's too concentrated. And there is a protocol, I think NIST has a protocol, we have a protocol up on our website that has some calculations where you can kind of estimate where the ideal dilution ranges are. But really you have to kind of dilute them down to there, then look at them, and then determine on your own, and usually run maybe, I usually like to run a series of dilutions just to make sure that some of the stuff that we're not seeing is, again, not compositional to the sample, and just to make sure it's not something that's in the sample that's giving us these, multiples of that single peak. And you can just basically what I do then is, if I see a single peak, then you can actually walk it up a little bit or increase your acquisition time to get a sufficient amount of particles to run a statistical analysis on.
Yeah, exactly. Yeah, you need to collect more data, right? Mm-hmm. Okay, so next question. How do you decide on the best matrix for digesting nanoparticle samples?
Normally what we do is we look at and see what, try to find what they will dissolve in. So if it's an acid, what acid will dissolve in. And then normally we just go high, we'll go high, we'll microwave, and then we'll just also instead of digesting it, dilute it down in the 2% nitric or whatever we're going to run it in through the instrument, and then just run an elemental analysis on each of them. And if they're equivalent, then we can just dilute down without having to digest. If not, we find out, what's the minimum point that gives us the right concentration. Do we only need to add concentrated acid, or do we need to actually microwave? And it's just depending on what's in the samples. So certain things we have experience with, or we'll look up, basically do a lot of literature search and kind of see what matrices we need to use for these. I can give an example for the iodine that we use in this. We initially we're trying to stay away from acids because we were trying not to volatilize any of the iodine, let it reduce down into I2, and then we just lose it. So that made us settle on base. And so then, we had to do some research, and a lot of people, from what we read, just basically said that tetramethylammonium hydroxide, was the best to use for this. And we just went with that. And again, we tried digesting the samples, and we compared it to some other techniques using that. We looked at concentration like UV and some other things. And then once we got, consistent results across, that was our sample matrix that we used for digestion.
Oh, thanks. I think that's a really good answer. Yeah, I would also recommend TMAH for iodine work. One more question. How are we doing for time? We're doing okay. How do you decide on the best internal standard for gold nanoparticle work in ICP-MS?
It's kind of hard, right? We don't really have a standard. We used to use the NIST. They used to have a reference size standard for gold, which again, they're sold out and they don't have anymore.
Mm-hmm.
That used to be our go-to, but now we kind of just buy nanoparticles from a supplier, and we do TEM on it to actually kind of get the size, and then we'll run gold quantitation analysis on this, and then estimate a gold particle concentration based on those two values. And that's kind of what we have to do now to determine our own concentration for these things. Just because like I said, we don't have a traceable standard, so we kind of have to characterize these solutions for ourselves and then use those characterized solutions.
Yeah, yep, and I highly recommend that everyone does that for sure. I think that's it for questions in the list here. If anybody has any more questions, feel free to pop them in. There is Matt's contact here. I guess I can reach out to Matt if you have any additional questions. Other than that, I don't see anything else coming up, Matt.
Mm-hmm.
Well, all right. Well, I guess I will end it then. Well, thank you very much, Matt. That was a really good presentation. I really like some of the work you're doing there. And if anybody has any additional questions, feel free to reach out to Matt or myself. I can connect you with Matt. This will also be recorded and available on demand at atomicwebinars.com, along with other webinars as well. And yeah, thank everybody for attending, and have a great rest of your day. And thank you, Matt, for presenting. It's really good.
Thanks for having me.
Thank you.